My name is Roberta Ness. I chaired the workshop planning committee. I'm the former dean of the University of Texas School of Public Health. We'd like to welcome you back to the Academy's workshop. The potential contribution of cancer genomics to information to community investigations of unusual patterns of cancer. I'll be moderating this final roundtable session. This session will synthesize all the information we've heard today and propose a path forward. And I just want to give you a very, very quick thumbnail synthesis for those of you who haven't been with us all day of what I believe we've learned up until now. It's been a very rich and exciting workshop with a variety of tremendous experts in their fields. So I think what we have heard is that certainly everyone agrees that childhood cancer rates are increasing. The reasons for that are unclear. Yet there has been considerable advance in a number of technologies and techniques applied to this field, including metabolomics, genomics, epigenetics, perhaps proteomics, which we haven't heard as much about, and biomarkers. And all of these, we've seen examples where there is considerable opportunity to learn from these more futuristic techniques. At the same time, we have heard that there are substantial challenges to doing this work, including the rarity of the cancers, the limitation in biomarker specimen availability, in part because children are small, and the opportunities for funding and for collaboration. Despite those challenges, as I say, there have been some very substantial leaps forward. And we've also heard that we now know some lessons about how to deal with communication to those who are potentially affected. And those strategies are tremendously important. So that having been said, I'd like to now introduce our speakers. Our four panelists will give their brief introductory remarks, and then we'll jump into a panel discussion at the end. We'll begin with Jack Taylor, who's a senior emeritus in the epidemiology branch in the epigenetic and stem cell biology laboratory of the National Institutes of Environmental Health Sciences, National Institutes of Health. Following Dr. Taylor, we'll have Logan Spector. Dr. Spector is a professor and chair in pediatric cancer research, the director of the Division of Epidemiology and Clinical Research in the Department of Pediatrics at the University of Minnesota. Next, we'll hear again from Trevor Schaefer, pediatric cancer survivor and the inspiration for Trevor's Law, an act that calls for periodic updates to the guidelines for investigating potential cancer clusters. Mr. Schaefer has continued his advocacy work in the executive director and founder of Trevor's Trek Foundation. Finally, Jerry Fagliano, clinical professor and chair of the Department of Environmental and Occupational Health at the Dornsief School of Public Health at Drexel University, will provide his remarks. Thank you, everyone, for being here today. Just a reminder to the audience, we encourage your involvement in the workshop. As you may have seen if you were here previously, we are taking questions from the chat. Please use the chat box on your website to ask questions or make comments. Please include your name and affiliation with your remarks. And I would now like to invite Dr. Trevor to begin his remarks and thank Dr. Taylor, did I say Dr. Trevor? Dr. Taylor, and all of those present. Thank you.
So hi, I'm Jack Taylor. I'm a molecular epidemiologist at NIEHS. The focus of my work has been at looking at environmental and genetic determinants of cancer, mainly on breast cancer. I have not done any work on pediatric cancers or clusters, but my work has broadly followed a lot of the themes that we've heard about today. First, in terms of working on, I did some of the early work looking at whether there was mutational specificity in tumors from environmental exposures, working on, for example, aflatoxin-associated hepatocellular carcinomas or arylamine-induced bladder cancers, and looking at the differences depending upon etiologic exposure. I've done a fair amount of work on the genetic determinants of cancer, looking at inherited genes, some of the early work on gene-environment interaction, which when we started thought was going to explain a lot of adult cancers. That certainly doesn't seem to have proven to be true. And in the last 15 years or so, I focused mainly on the epigenetics, both of epigenetic changes induced by exposure and whether epigenetic profiles are associated with cancer risk. So we did some of the early work that you heard about earlier today about smoking-associated epigenetic changes, both in adults and in children, looking at alcohol exposure and epigenetics changes, looking at lifestyle exposures and epigenetic changes. And we've also, in breast cancer, done large studies looking at whether whether we can detect changes in the epigenome years prior to someone developing breast cancer. One of the things that I think is most interesting about epigenetics is that you can start to pull different information from sets of sites across the genome that inform you about exposure and disease risk. And in particular, you can use epigenetics as an estimator of biologic age, and that age can be different than your chronologic age. And we are seeing how exposures, environmental exposures, can make you biologically older than your chronologic age, and investigating how that risk or whether that risk, whether that change is associated with cancer risk and risk of other diseases. So this has been an incredibly fun morning for me, hearing new things in terms of mutations, epigenetic changes, but it's a new area in terms, for me at least, in terms of childhood cancers.
I think it's my turn to speak. So I'm Logan Spector. I'm a professor at the University of Minnesota and a childhood cancer epidemiologist. I am the former chair, immediate past chair for epidemiology with the Children's Oncology Group, and currently chair the Childhood Cancer and Leukemia International Consortium that Dr. Wiemels mentioned. You are, by the way, welcome to go to our website, www.clic.ngo, to learn more about our consortium, which pools epidemiologic data from case control studies of childhood cancer around the world in order to come up with answers about causation. I very much appreciate this panel and that the National Academies are trying to bring cluster investigations into the molecular era. I think that there's a lot of potential there. And having personally been involved in several cluster investigations, not as a primary investigator, but as a consultant. I certainly have suggested that studying the tumors, as well as the both affected and unaffected community members' genomes, could be a productive approach. If not to nail down a causative exposure, at least to show that the cases that occurred during a time of alleged increased incidence resembled or did not resemble reference data sets. That alone could be a useful endeavor. I'm also, having been part of these investigations, and I'm also keenly aware of the sometimes delicate or difficult relation between the scientists or public health workforce that conduct these investigations and the community that longs for answers. So I very much appreciated that discussion as well, and I'm looking forward to the discussion that we're about to have after we hear from our last two panel members.
Am I next? My name is Trevor Schaefer. I started Trevor's Trek Foundation, co-founded with my mother after my diagnosis of brain cancer when I was a child. And we started that organization to look at issues surrounding childhood cancer, primarily regarding the environment and toxic exposure. We worked for 7 years to get a law passed called Trevor's Law, which addresses cancer clusters throughout the United States and was signed by President Obama on June 22nd, 2016. Our work now with Trevor's Trek Foundation revolves around how we can bridge the communication gap primarily between communities and our federal, state, and local health departments when addressing cancer cluster concerns. We're doing several things to hopefully help complement that work, and from childhood cancer data collection to mapping to identify childhood cancer clusters, to work with scientists out of the University of Arizona, Dr. Mark Witten and Paul Shepard, experts in childhood cancer cluster investigations and dendrochronology, the study of tree coring, and also look at ways that we can focus on remediation efforts to introduce products to help with that. So very much looking forward to this ongoing conversation to discuss how we can best implement the new cancer cluster guidelines and Trevor's Law to help give our communities a voice and a place to go to express their cancer cluster concerns, and have our local and state and federal health departments respond to those in a timely, honest manner.
Thanks, Trevor. And good afternoon, everyone. My name is Jerry Faliano. Yes, I'm now at Drexel University, but I spent most of my career in state government. One of the things that I was involved with was that I led the investigation of the childhood cancer cluster in Toms River, New Jersey, which has had some reference over the course of the day and in the materials. That investigation started about 25 years ago. It was finished about 20 years ago. So it's been interesting for me to listen to the tremendous progress that's been made in understanding exposure assessment, great development in tools for understanding exposures and how they impact the genome, So this has been a really fascinating day for me. I had also been involved in the 2013 version of the CDC's guidelines, and it was interesting for me to look at the 2022 version and see the kinds of changes that have been made. Most of the structure is more or less the same, but there have been some interesting updates. So I listened today, putting myself back in the role, my former role as a state health department scientist and official, someone who was responsible for responding to communities and communicating with communities. So I would echo what Logan said about how interesting the session prior to this one was in terms of thinking about communication and the two-way communication and obligations of state officials when it comes to discussing these really complicated issues. I guess I just want to comment on one or two things from today. What struck me about listening to the sessions today is sort of contrasting what a scientist has to do who's working in the state agency or working with registry data, and needing to respond to communities. One of the important features of a cluster is that they don't get recognized really until well after the exposure windows of concern or of interest. And in the case of the Toms River study, which was not in a very small community, we had to think about reconstructing exposures over a 35-year period, dating back to the early 1960s through the middle 1990s, given the case grouping. So that was because it takes a while for as had been pointed out, the reporting of cancer cases to get to a registry, for integration of all the data, for the application of whatever analyses are done to recognize the fact of a cluster, and just the simple time it takes to accrue a sufficiently large number of cases to recognize the cancer and the cluster rather, and be able to sort of characterize it in time and space. So I think that creates such huge challenges in thinking about how do we apply these modern tools when even a cluster that's recognized today is still going to have to look back very long period of time in segments of time for which we really don't have good information. Again, the promise is there that we may be able to reconstruct exposures. So I'm again looking forward to the further development. I don't think we're quite at a place where what we know now is applicable to changing the guidelines or integrating into the guidelines in any way. But I'm very interested in hearing what other people's perspectives might be on it. So thank you.
Sorry, I was on mute. So I want to thank all of you for your comments, your insights. Very, very interesting and useful. And I now want to invite all of our speakers to the panel discussion. We have about 30 minutes, so quite a bit of time to talk about what we've heard today, and get their comments, their suggestions. And moreover, again, I remind all of you who are in the audience to please participate using the chat box. So let me just start out. I earlier indicated what I had learned from today, and I'd just like to ask all of the panelists now what you have learned, and particularly what you've learned about the investigation of cancer clusters? Any of you? Jump right in.
What I know about investigating cancer clusters is from, being a consultant. Today, I did learn very useful things about applying molecular methods, but to my knowledge, they haven't been employed yet in a cluster investigation. I think that, what I've learned is just how hard they are to do beyond using registry data. The registry data is, available. It's the first, go-to data for any health department, which is usually who's conducting a cluster investigation. That's where they would first go. But when it comes time to do, a quote live study requiring, people to engage with the researchers and give their information, there's a lot of fall off from the roster of everybody that's in a registry to those who are able to be found, willing to talk with the researchers or give useful information. There is also sometimes a mismatch between what the community wants and the people who maybe initiated an investigation. I will just give you one example from Minnesota. There's a cluster of mesothelioma up in the Iron Range, and it's due to one of the minerals they're trying to mine has asbestos in it, and there was a community member who got it as far as the legislature to fund a study of whether this mining was related to mesothelioma. But they had a real hard time actually engaging anybody to respond, because mining, the industry was the heart of the town, and it turned out that people did not care to have the economic heart of the town questioned. And so I think that we're starting from the point that everybody would want to know about an environmental hazard, but I don't think that's necessarily true. So anyway, we talked a lot about science, and there is definitely a social aspect and maybe some social competencies that cluster investigators need in order to make these investigations more fruitful.
Thank you. I'm really going to ask each of the panelists to briefly jump in and answer this question.
So one of the things that I found most intriguing was it wasn't cluster specific, but it was childhood cancer specific. The presence of a virus sequence within tumors and the ability to separate out different etiologies of tumors based on whether or not there was viral sequence there. And also there was a parsing as well by prenatal smoking and chromosomal abnormalities. Both of those findings really emphasize something that multiple speakers brought up about the developmental origins of cancer, that we've got an exposure window that doesn't start at birth, it starts before that, which means that both the mother and the father's exposures and exposure, particularly during the prenatal period, may be critically important to consider, and so that was a really nice emphasis that was brought home in a molecular way for me on this.
I just want to jump in briefly and add to Jack's comment, which is an outstanding one. I've worked a lot in my career with very, very large European pregnancy cohort studies, including the Danish National Birth Cohort, Norwegian Birth Cohort, Swedish, et cetera. There's a British one, but there are a number of them. And they do collect biospecimen data from mothers, but also in some cases from the babies, the neonates, and then children thereafter. And I think that, and unfortunately, to my knowledge, none of them have been particularly focused on this area of research. And I think that there's a profound opportunity, perhaps, to involve those very rich data resources in this area. Okay, Trevor, Jerry.
I wanted to what stood out most to me, and it actually ties in very well with what Dr. Spector was saying about community engagement in studies, is that something that in the last session that Dr. Lash had commented on, and Dr. Ramirez, who was in the session, also was speaking to, and that is community communication with our health departments, and maybe sometimes communities not knowing where or how to make that connection. And I think that's a crucially important piece to what we're discussing and how we can move forward. And what I talked about that I think would help solve that issue is establishing a community liaison, individuals throughout the country who could help bridge that gap between the communities and between the health departments. Make sure that the data is being shared correctly. Make sure that the community engagement is there when performing studies in areas. So I think that's a very important piece, and focusing on establishing those individuals in different regions of the country, liaisons that can help with that, I think it is the best solution to solve that problem.
So I guess one of the thoughts that came to me as I was listening to the sessions today, sort of reflecting on my experience with the Tom Zerber investigation and also the Woburn investigation, where there are two studies in two different communities looking at a drinking water exposure pathway. Both find somewhat strong associations between maternal exposure or maternal access to contaminated well water during the pregnancy period and later risk of leukemia in the children. Both studies, have a common identify a common time window. But these two studies looked at very different contaminant mixes in those two places. And so what intrigued me about the tools that were being talked about today was this promise of potentially identifying ways that there's common mechanisms could be discovered through, even if there's different kinds of exposures, if they're operating through common pathways. If I understood what was being talked about today, I think that seems to me a possibility to tie together, insults from environmental exposures that may be different but may operate in through similar mechanisms. And so that was the thing that struck me probably the most out of the conversations today.
Okay, I'd like to acknowledge a question or a comment from one of our participants in the chat box. This is an individual who's an RN who says that advanced research resources should be connected with the communities. Seems to be a disconnect there, especially for prevention and follow-up. Sadly, communities sometimes don't have a high degree of trust. And I think that's a really salient comment. And I'd like to ask any of the panel participants to just advise us about that.
Yeah, Agnes, thank you for your participation today in all of your work. It's a big issue, and it's something that I've been talking about for a long time. And what we're trying to do is what Agnes mentioned about communities don't trust the health departments. They don't trust the CDC, although individuals I'm working with there are very strong and very compassionate and want to do the right thing. And what we're trying to do at Trevor's Trek is we're trying to help reinstill some trust with communities in the federal government. But in order to do that, the federal government and state and local health departments, they need help. They need help from organizations outside of the government to come step in and take some of the burden off, because they can't do it all. So what we're doing today, the publishing of the new guidelines in December and this workshop, these are all great steps to improving that and making sure that happens, and working with organizations like Trevor's Trek and like others out there that are like-minded and just want answers and are willing, and community members who want answers and are willing to do what they need to do to help with that. It's very important. So trying to reinstill that trust with the federal government is huge for that, because there has been some, to put it lightly, some lack of oversight over the years. So we definitely need to work on that.
I would agree with Trevor that having community organizations engaged in what is ultimately done as far as investigations go in a community is really important. And of course, that was talked about earlier today as well in great detail. I think hoping that the state or the CDC would gain the trust of a community is probably not maybe the right way to frame it, but I think what is important is that the individual scientists who are working with families and with communities need to do the work to gain trust for at least their own work with that community, and that does take time and it takes effort. You often start behind the 8 ball when it comes to a level of trust with you, but it's really incumbent on every investigator to make that effort and to demonstrate their commitment and their integrity. Again, I think that's what they can do. They can't necessarily move the entire perception of the federal government or someone's perception of the government in general, but at least they can build trust with themselves.
One of the things that strikes me is that, and we've heard this from Trevor, we've heard it now from Jerry and from others today, the conversations that are being had around communicating with communities are kind of micro conversations. So they are conversations with the affected community. There's certainly echo conversations, i.e. conversations that scientists are having with each other. But I think that there is a really tremendous limitation in reaching out to the public in general and letting them know what to do, as Trevor noted, about potential cancer clusters and how do these get investigated, and how can we deal with this, and how can we deal with our concerns and worries? Because I think there are an awful lot out there. So that may also be a take home for the CDC et al.
Just one comment, Roberta. The timeliness of the response and getting information back is a particular challenge. The studies that we do, certainly involving genetics and epigenetics, are years after the collection of the samples. And so it really becomes problematic in terms of thinking about any sort of timeliness in terms of returning information. The environmental monitoring things may have a quicker turnaround. And I think we have to think about what things can we get back fast, and provide to people, what things can't we? And I also worry about the much of the data, at least epigenetic data, that we collect, for example, is very difficult for me to interpret, and providing it to someone, to a patient, and then essentially asking their physician to interpret, becomes a real problem. So I think the patient has a right to know, but it really is a difficult thing when we don't understand it ourselves, we as researchers.
Yeah, there's also a profound difficulty with the disconnect between the timeline for cellular biology, cellular biologic studies, and epidemiologic studies. And one of the things that's concerned me for a very long time is the way that the media handles scientific information, particularly from those non-full human sources. So that what it does is it raises the expectations of the public that a particular new technology or particular finding may have tremendous impact. But to get those studies then into epidemiology, to get them funded, to get them accomplished, takes years and years and years. And that erodes the trust of the public. I don't know if any of the rest of you would like to comment on this. I think Jack is bringing up a really critical point.
Roberta, are you talking about the so-called CSI effect, where the public thinks that molecular tools can be employed in the space of an hour TV show?
You got it.
Yeah, but that's probably not scientists' fault, but the screenwriter's fault.
Well, I'm saying it's the media as well, but it's also the fact that many scientists have drawn kind of a hard line between science and advocacy. So we don't get out and spread the message. We don't get out and disseminate, I think, more well curated information. So I think it's both. Okay, so I want to move to a next question. Megan Whale Latshaw posed a question that I'm just going to expand on a little bit. It's a very good one. What do you think are the best strategies to assess and respond to potential pediatric cancer clusters in communities, given what you've heard today? Maybe we can start with Jerry.
So, I mean, I think the idea of creating national registry is tied to systematic collection of biological specimens that can be banked. I think those are all the right things. I don't think we're at a place where this can be necessarily implemented yet, but in the next couple of decades, I think the those kind of systems in place can be extremely helpful in responding to the situations where it may be useful to employ those tools. And one thing I wanted to say is that the guidelines, the current guidelines or the new guidelines, as well as the old ones, funneled the more intensive work into situations where you had a true cluster, meaning there was an incidence greater than expected, where there was a likely unusual or unique exposure that could explain a localized cluster, and where there's at least a reasonable number of cases that could be studied to test a hypothesis. So I think given those kind of relatively rare circumstances where a study might be able to be done, I think using those sorts of systems that might be in place over the next couple of decades, there could be much greater capacity to look for past exposures, or to look for associations between past exposures and groupings of cancers that fit together in a realistic way.
Other comments on this question?
I mean, I'll echo what Dr. Faliano just said. Having the National Cancer Registry, Childhood Cancer Registry, and being able to look at it clusters systematically and with population-based data is absolutely key. However, I share, I guess, skepticism that it can be implemented in real time. There's simply a process of abstracting cancer and deduplicating and et cetera that cannot be compressed. So it will be honestly very difficult to make this a real-time reactive system.
Okay, and that segues nicely to a comment made by Stephanie Foster, who is one of the key staff that supported our committee's work. And she says, what was reinforced for me today is the importance of a collaborative approach to examinations of unusual patterns of cancer. Work is still needed in omic science and in the development of processes to advance data availability and data interoperability from multidisciplinary inputs such as genomic cancer registries, community participatory science, and electronic health records, establishing collaborations with academic researchers, public health practitioners, and impacted communities can be a pathway to accomplish this in the future. And I think that's a beautiful synthesis of much of what we've heard today. I'm going to ask a question, but this may also be a very good question for all of us to consider, really earlier panels. From Emmanuel, I am going to totally mess up this name, Netkop, who says, why is more information on cancer and environmental causes not made available to the public as they emerge by government and private institutions, especially, and this is not, when it first appears in peer-reviewed journals? Any comments about that, or perhaps we can hear some?
Well,
Let me.
can I just say one thing about that?
Yeah, thank you, Sharon.
Since you said it was open to anyone. The thing that's really surprised me all day is the word NIH grant hasn't actually been mentioned. And so for the public that's listening, most of our research is funded not through the CDC or some of the other organizations that are participating today, but through the National Institutes of Health. And making your data available to the public is not a common measure of success. I just have to be honest. We're judged on how many scientific papers we publish, how many people we train, and in some ways on the impact did our work turn into new cancer therapies or new medicines. But it's really only in the last few years that there's been quite a change in working with the public as stakeholders and partners, I should say, sorry, not stakeholders, as partners at all phases, including making this information known. And there's a new grant organization, not new anymore, but probably about 12 years old, called the CORI, which is about patient-centered research. And that group funds, that organization is also from the federal government, funds more research that emphasizes it. But I do think a fundamental issue has been that that's not what we've needed to show in order to get funding for our research. And I think it's only recently that has become more part, and really I think it's still a small part, of how our applications for research funding, which are called grants, are reviewed.
Thank you. If there's anyone else from earlier in today's sessions who would like to jump in on this one, I'd be most appreciative. Or the current panelists.
Well, as long as we're talking about funding, cluster investigations are, as far as I know, almost invariably conducted by a health department or the CDC, not academics. And I don't think that the health departments have the funding and may not have the direct expertise to employ the molecular techniques that we talked about. So, just nuts and bolts here. If those resources aren't made available to health departments or CDC, if they're called in, then these are never going to get incorporated into investigations. And again, did suggest this in Pennsylvania, which is looking into whether fracking and fracking waste could be a cause of childhood cancers. And although they did get a grant or money to fund the study from the state, by the time it was, reached the external advisory board for advice, my advice was to invest in the molecular omics. The money had already been spoken for, and it just wasn't there. So there needs to be an intentional process of funding incorporation of omic science into cluster investigations.
Just a comment on the environmental determinants of cancer in general. It's a really tough nut to crack. And the genetic determinants of cancer have been much easier because essentially all you need is a blood sample at any point in time from the person, and you can gather all of that all the information you need in a case control way. Determining the environmental things means you have to construct this whole history. And I agree, we have to put the information out there. Publication is the way we do it, and news resources pick that up. More resources for the environmental determinants of cancer is critically important, because we know it's a box that accounts for a large chunk of cancer. We know what genetics accounts for. We know environment must be the rest. but it's been a really intractable box to split open and figure out.
Yeah, Jack, let me just ask you and follow up to that. When you say it's been a tough nut to crack, just either some examples or a little bit more specificity on that?
Yeah, so my experience has mainly been or the example I'll bring forward is in breast cancer, where we now know through a lot of consortia efforts about the genetic determinants, the rare polymorphisms, the common polymorphisms. Most of the genetics of breast cancer we have figured out. There isn't a lot of latitude left there. But the ability to work internationally to figure out the environmental determinants is harder, because we have to ask people questions. We have to try to reconstruct all of this exposure history, which may all go all the way back to include prenatal exposure, which an adult woman probably doesn't know what her prenatal exposure was. And so it's very hard to get the good epidemiologic data that we need to inform that in the sample sizes that we clearly need in order to inform that. So in that way, the genetics is much easier than figuring out the environmental part of the story.
Yeah. What I'm hearing from you is that there needs to be a level of intentionality prior to launching the study, such that environmental exposures are captured, not just the omic, not just using omic approaches, but actually using the environment itself, whether it be from dust or water or air or whatever it is, because otherwise you can't validate the markers. And that we really haven't had that in any of these large studies that I mentioned previously, and that's absolutely the case. I mean, unfortunately, we had the opportunity, we lost the opportunity in the failed National Children's Study. That was the chance to do it. Hopefully that'll come around again at some point in the future. But yes, absolutely. Let me just ask, we have somewhat more limited time now, about research gaps that really struck you today. And, if you were going to do another workshop, what would you like discussed? Any of you?
Well, as far as gaps goes, I'm as knowledgeable of childhood cancer epidemiology as anybody, and we have generations of epidemiologic studies, case control studies that might have gotten germline DNA, might have a methylation profile from a blood spot, might have good high quality questionnaire data, residential history, et cetera. But what they don't have is the tumor. There are precious few studies that have the trifecta of good exposure data, good germline and pretreatment samples, and then the tumor. And so if we really want to close the loop and be able to connect exposure to some sort of fingerprint in the tumor, then we would need a new generation of studies that have those.
Trevor, Jerry, Jack?
We need to, what I would like to see is a discussion about kind of what I feel like a broken record here a little bit, but establishing that liaison in different regions of the country to connect our communities with our health departments, with our federal government, when addressing cancer cluster concerns, whether it's a lot of universities who have a strong focus on environmentally related issues and connecting it with cancer. A lot of very, very smart, qualified individuals who can really help assist with that effort and create that communication. I've talked to and we work with a lot of individuals, whether it's individuals or it's representatives, doctors from universities who care about these issues, and they want to know how they can get involved and how they can help with this effort. So I saw a comment that Grace made as well, that says MDs that should get hired, professionals to hire to do an environmental exposure history. And I think that's something that we should consider, because it is a big task to ask for that assistance, and so they should be compensated for that effort. So I think there's plenty of funding in this country to be able to do something like this, and this is a crucially important issue, and it definitely deserves that attention.
Thank you. I want to use the last few minutes that we have to jump to a final question. And that is, 10 years from now, if there were no barriers, what would the investigation of cancer clusters look like in your mind?
No barriers whatsoever, because there are multiple barriers that would have to be overcome. But we would compress the time to register a patient to the minimum. We could then have maybe not real time, but we could have investigations when there appears to be a statistical anomaly suggesting a developing cluster within weeks or months. And then we have to, but if we have that, then this is different than the usual paradigm of community concern starting a cluster investigation. Instead, the investigators would be coming in and saying, we think you have a cluster. I'm not entirely sure how well that would work. But that is sort of what we've been talking about. And then we would conduct an epidemiologic study like we normally do, just maybe a lot grander methods and a lot more quickly with fresh memories, rather than coming in years later to try and reconstruct history. That's what it would look like, but I do think there are substantial barriers to that.
I think Logan described it perfectly well. One thing that I think has been touched on in this during the sessions today was also the acknowledgement that the investigation is happening in a community that has a variety of concerns. And I think in parallel, the health agency needs to be working and coordinating closely with environmental agencies to understand whether there are actionable issues in the environment that people are also concerned about in parallel. Whether you've drawn the connection or not is in many ways not the point. But if there are exposures that ought to be taken care of, I think demonstrating that in parallel with whatever investigation you're doing goes a long way toward helping to build trust that we talked about earlier, but also, maybe ultimately an action that will down the road will be shown to prevent future cases. So I think doing that kind of effort in parallel with these kind of investigations is also really important. So I would build that in a very strong way.
Okay, well, I think that our time is almost up here for this session. I'd like to thank everyone for attending today's workshop in general, which was convened by the National Cancer Policy Forum in collaboration with the Roundtable on Genomics and Precision Health. We're deeply appreciative to our exemplary speakers today for their rich discussions. It really was very illuminating. Special thanks as well to the Planning Committee. Thank you personally from my heart, who spent countless hours designing this workshop. And I would also like to thank the staff of the National Academies who I've worked with many times before on other committees. Their level of support, their level of involvement, of intellectual input, of integrity are really unsurpassed. Lastly, I'd like to remind everyone that speaker presentations are posted on the project website, and the webcast itself will be archived in approximately 10 days. Thank you again for joining us for this important workshop. I hope you enjoy the rest of your day.